Protein structure. There are 4 levels of protein structure. The shape of a protein is super important because, as I mentioned before, if you don't have the right shape, then the protein won't work. So for example, hemoglobin, if you have one amino acid change, then the protein is shaped differently, and then hemoglobin doesn't carry oxygen quite as well as it did before. So the shape of a protein is super, super important. So we're going to spend a lot of time talking about protein structure. There's a primary structure. Primary means one. And a secondary structure. And a tertiary structure. And finally, a quaternary structure. They do not all have a quaternary structure, but they all do have primary, secondary, and tertiary structures. So let's talk about that. Primary structure. The primary structure is just which amino acid comes first and which amino acid comes second. So the amino acids bonded together by peptide bonds. So here's the primary structure. This amino acid, and then that one, and then that one. It's the order or the sequence of amino acids. So for example, here's a protein. The primary structure is the specific sequence of amino acids in the protein. So here's the amino acid lysine, and after that comes valine, and after that phenylalanine, and then glycine, and then arginine. So the order of amino acids is super important, and each one of these is an amino acid. Let's say the protein was messed up and it didn't have that amino acid, then the whole shape might be messed up. This protein is also called a polypeptide. So when we talk about polymers and monomers, the polymer is a polypeptide. The monomer, I wish it were a monopeptide, but the monomer is called an amino acid. That's this. So which of course is a special polymer. So there are several different polymers. There are polypeptides, which are polymers of proteins, and there are polysaccharides, which are polymers of carbohydrates. So right now we're talking about proteins, so we're talking about polypeptides. You can almost use the word polypeptide interchangeably with a protein. It gets a little bit different when we get to the quaternary structure, and I'll show you that when we get there. Secondary structure. This is the three-dimensional folding arrangement of a primary structure into coils and pleats held together by hydrogen bonds. So let's take a look at that. There are 2 examples. There's an alpha helix. And so what you'd have to picture here is an amino acid connected to an amino acid connected to an amino acid. And this chain of amino acids tends to coil because there's an atom sticking out here that's attracted to an atom sticking out here. So you get hydrogen bonds between the different parts, and it makes this coil. You can have the same thing in this beta pleated sheet, and so they'll also be hydrogen bonds here, and it'll make the whole thing kind of form this sheet. And so the blue dots are supposed to be hydrogen bonds. So here's another picture of an alpha helix and a beta pleated sheet. So here's the amino acid, and it forms this coil because there's a hydrogen bond between these two. So this amino acid is not adjacent. It's not right next to this amino acid, but when they coil, when the whole thing coils, they end up being close together. So it's not closely covalently bonded. That would be this way, but when it folds, it ends up being close together. And then there's a hydrogen bond here, and so on. And so if you remember when we did the way amino acids are, it's nitrogen to carbon, nitrogen, carbon, carbon, like that. And so here's maybe the central carbon, and this would be the COH group, and here's the nitrogen, and that's bonded to another carbon. And so that would be the next amino acid. Here would be this amino acid. Anyway, this one is attracted to the one way down here. So here's the next one. Here's the next one. So, whoops. So the carboxyl group here is attracted. It has a hydrogen bond with the amino group way up here. So that's actually 1, 2, 3 amino acids in between them. Here's a beta pleated sheet, and so they fold over and they make this kind of funky sheet. So the secondary structure is due to hydrogen bonds. The tertiary structure is when the whole thing folds over on itself. So the tertiary structure forms when a protein chain or a polypeptide folds over on itself due to the interactions of functional groups. You can have hydrogen bonds, ionic bonds, disulfide bonds, which means a sulfur bonded to a sulfur. And so let's take a look at that. So get this out of the way. So you can see you've got this beta pleated sheet here, and then it forms an alpha helix over here. And so the way this works is that this part might be bonded here by a disulfide bond, let's say, and you've got, of course, the hydrogen bonds that are connecting the loops here of the alpha helix. And then there might be an ionic bond here to here, and there might be another hydrogen bond here, and this one might be a disulfide bond here. So that would be the tertiary structure when you have this long chain and it folds over on itself. So that's an alpha helix and a beta pleated sheet, and those are all part of this one polypeptide chain. This three-dimensional structure is unique to each protein and important for proper function. So if the structure changes, it doesn't work anymore. Folding over is driven by R group interactions. So if you remember what R groups are, you've got a carbon, and it'll be bonded to this carbon, then I'll have the COOH, pretend that's an H, and this will be double bonded to an O, pretend that's a double bond. And then you've got your amino group here that has the H's sticking off of it. And then there's this R group. So it's the R group, whoops, it's these R group interactions that we're talking about. So here's an R group, and it's attracted to this other R group, and here's another one, sulfur to sulfur. And here's another one that's actually, these are pretty far apart on the chain, but once they fold over, this amino acid is very close to this one. So here's the R group that's attracted to the other R group. Quaternary structure is the last structure. So some amino acids don't even, some proteins don't even have this. Some proteins finish with this tertiary structure. Quaternary structure is just in some proteins. So it's when different protein chains or different polypeptides join together, you get this quaternary structure. And so here's a polypeptide chain, and another polypeptide chain, and another polypeptide chain, and another polypeptide chain. Each of these polypeptide chains has formed alpha helices and beta pleated sheets, and they've also folded over on top of themselves, and then they've joined together to these other polypeptide chains. So once you have these four polypeptide chains together, now you can call it a protein. There are some proteins that are made of just one chain, and so that would finish with the tertiary structure, but this protein has four polypeptide chains to make the protein. So you can use the term polypeptide chain almost interchangeably with protein, but they're a little bit differently in that if you have a quaternary structure, you have more than one polypeptide chain to make your protein. So an example is hemoglobin. There are actually four polypeptides bonded together to make hemoglobin. So here's one, and here's another, and here's another, and here's another. There are two alpha chains and two beta chains, and they have this heme group attached to them. But there are these four different chains that make up the protein. And so the primary structure would be which amino acid is here, bonded to the next amino acid, bonded to the next amino acid. And then alpha helices would give you the next level. And now that these things have folded over on top of each other, that's the tertiary structure. And then that you have 1, 2, 3, 4 chains gives you the quaternary structure. Here's another example. This is collagen. There are 3 polypeptides twisted together. So you can see the alpha helices in each of these 3, and then the 3 are twisted together. So that's why collagen is so strong. You have these 3 cords of proteins actually twisted together, and it makes it strong, kind of like twine. So here's the protein structure. The amino acid sequence is the primary structure, which amino acid is first, which is second, which is third. And then they make these alpha helices or beta pleated sheets, and that's because of these hydrogen bonds between different parts. And these are still fairly close together, so that's the secondary structure. When these things fold over on top of themselves, that's the tertiary structure. Some proteins are done right there with the tertiary structure. And then the quaternary structure is when you have more than one polypeptide chain. And you're going to miss this video because they never work. for me. And we'll practice a lot of this in class. Okay, and that is it.